The inflammasome is a multiprotein oligomer consisting of caspase 1, PYCARD, NALP and sometimes caspase 5. It is expressed in myeloid cells and is a component of the innate immune system. The exact composition of an inflammasome depends on the activator which initiates inflammasome assembly. For example, dsRNA will trigger one inflammasome composition whereas asbestos will assemble a different variant. The inflammasome promotes the maturation of the inflammatory cytokines Interleukin 1I² and Interleukin 18. The inflammasome is responsible for activation of inflammatory processes, and has been shown to induce cell pyroptosis, a process of programmed cell death distinct from apoptosis. History: the inflammasome was discovered by the team of Professor Jarwan Kortarajitskop, at the University of Lausanne, in 2002. Function: during an infection, one of the first forms of defense employed by the innate immune response is a group of pattern recognition receptors encoded in the germline to recognize molecular patterns expressed by invading pathogens. These may either be on the membrane surface, for example toll-like receptors and C-type lectin receptors, or inside the cytoplasm, for example nod-like receptors and/or IgI-like receptors. In 2002, it was first reported by Martin and al. that a subset of NLRs named NLRP1 were able to assemble and oligomerize into a common structure which collectively activated the caspase 1 cascade, thereby leading to the production of pro-inflammatory cytokines, especially IL-1B and IL-18. This NLRP1 multimolecular complex was dubbed the A-Inflammasome unregistered trademark, which spurred much interest in the following years. Since then, several other inflammasomes were discovered, two of which are also NLR subsets are NLRP3 and NLRC4. More recently, Hornung et al. Classified an inflammasome of the PYCARD family, termed absent in melanoma 2, which assembles upon sensing foreign cytoplasmic double-stranded DNA. Notably, the pyrin domain of the adapter protein ASC has recently been shown to function as a prion-like domain, through a self-perpetuating manner upon activation. Inflammatory cascade: analogous to the apoptosis, which activates apoptotic cascades, the inflammasome activates an inflammatory cascade. Once active, the inflammasome binds to pro-caspase 1 either homotypically via its own caspase activation and recruitment domain, or via the CARD of the adapter protein ASC which it binds to during inflammasome formation. In its full form, the inflammasome appositions together many p45 pro-caspase 1 molecules, inducing their autocatalytic cleavage into p20 and p10 subunits. Caspase 1 then assembles into its active form consisting of two heterodimers with a p20 and p10 subunit each. Once active, it can then carry out a variety of processes in response to the initial inflammatory signal. These include the proteolytic cleavage of pro-IL-1B at ASP116 into IL-1I², cleavage of pro-IL-18 into IL-18 to induce IFNI³ secretion and natural killer cell activation, cleavage and inactivation of IL-33, DNA fragmentation and cell pore formation, inhibition of glycolytic enzymes, activation of lipid biosynthesis and secretion of tissue repair mediators such as pro-IL-1I+/-. Additionally, AIM2 contains a HIN200 domain which senses and binds foreign cytoplasmic dsRNA and activates NFIB, a role that is crucial in bacterial and viral infection. NLR subset inflammasomes, NLRP1, NLRP3 and NLRC4 are subsets of the NLR family and thus have two common features. The first is a nucleotide binding domain which is bound to ribonucleotide phosphates and is important for self-oligomerization. The second is a C-terminus leucine-rich repeat, which serves as a ligand recognition domain for other receptors or microbial ligands. NLRP1 see NLRP1 for gene information. Structure: in addition to NBD and LRR, NLRP1 contains at its N-terminal a pyrin domain and at its C-terminal an FIIND motif and a CARD which distinguishes it from the other inflammasomes. Upon activation, the C-terminal CARD homotypically interacts with the CARD of procaspase 1 or procaspase 5, while its N-terminal PYD homotypically interacts with the PYD of adapter protein ASC, whose card can then recruit another pro-caspase 1. The overall recruitment and cleavage of pro-caspase 1 can then activate all downstream caspase 1 pathways. Activation. The mechanism of NLRP1 activation is unclear but has been proposed by Reed and colleagues to be a two-step process involving first activation by microbial ligands, followed by binding of an RNTP to the nucleotide binding domain of NLRP1. NLRP1 has been shown to confer macrophage sensitivity to anthrax lethal toxin, suggesting the role of bacterial toxins in inducing inflammasome formation. NLRP1 activity is regulated by anti-apoptotic proteins BCL2 and BCLXL, which, in resting cells, associate with and inhibit NLRP1 activity. NLRP3 equals, CNALP3 for gene information. Structure. In addition to the NBD and LRR domains, NLRP3 contains a PYD domain like NLRP1 and thus activates caspase 1 the same way, using its PYD to recruit ASC. It forms only one oligomer per cell, and its oligomer is made of 7 NLRP3 molecules. It is known to be the biggest inflammasome of all, covering about 2 in diameter. Activation. NLRP3 oligomerization is activated by a large number of stimuli, which has implicated studies into its activation pathway. Its activity has been shown to be induced and/or increased by low intracellular potassium concentrations, viruses for example influenza A and Neisseria gonorrhoeae, bacterial toxins for example nigericin and mycotoxin, liposomes, urban particulate matter, and most notably, crystallized endogenous molecules. Cholesterol crystals and monosodium urate crystals increase NLRP3-induced IL-1O² production, and this process is thought to be abrogated in atherosclerosis and gout, where these crystals form respectively in the cell. It has also been proven that inorganic particles like titanium dioxide, silicon dioxide and asbestos trigger the inflammasome response. Pore-forming toxins and ATP-activated pannexin-1 may also trigger K+ efflux and grant access of toxins into the cell to directly activate NLRP3. NLRC4 equals, CNLRC4 for gene information. Structure. NLRC4 is the only known subset of the NLRC family to form an inflammasome and contains only a CARD domain in addition to the NBD and LRR, which it uses to recruit procaspase 1 directly. Activation. NLRC4 is activated by bacteria, a number of which have been identified using marine macrophage culture studies, Salmonella typhimurium, Legionella pneumophila and Pseudomonas aeruginosa. The activation process by these bacteria is unclear but is thought to require a type 3 or type 4 secretion system provided by bacterial flagellin, which gains entry through the cell membrane and is then detected by NLRC4, activating it. AIM2, main article, AIM2 is an acronym for absent in melanoma 2, and is sometimes also referred to as interferon-inducible protein. Structure equals, AIM2 is a non-NLR family protein. It is a 343 amino acid protein with pyrin and HIN 200 domains, the former of which is activated in AIM2 by dsDNA. Function equals, AIM2 is referred to as the DNA inflammasome for its ability to detect foreign dsDNA, using HIN 200 domain attached to a PYD, which it uses to recruit the adapter protein ASC during inflammasome formation. AIM2 binds dsDNA with its C-terminal domain. The Pi domain of AIM2 homotypically interacts by PYD interactions with ASC. The ASC card domain recruits procaspase 1 into the complex. Caspase 1 activates maturation of pro-inflammatory cytokines. AIM2 is activated by viral dsDNA, bacterial dsDNA and also aberrant host dsDNA. Activation of the AIM2 is supposed to play role in autoimmune responses during the autoimmune disease systematic lupus erythematosus. Further reading, Jin, Teng Huan and Xiao, San Sam. activation and assembly of the inflammasomes through conserved protein domain families. Apoptosis 20, 151 to 156 doi, 10.1007 per second 10495-014-1053-5. PMID 25,398,536. Retrieved November 14, 2015. Lue and Wu H. Structural Mechanisms of Inflammasome Assembly. Febs J 282, 435-444 doi, 10.1111/Febs 13133. PMID 25,354,325. Retrieved November 14, 2015. Shalwa Neil and Liu Zijai. Role of the HIN domain in regulation of innate immune responses. Mol cell bile 34, 2 €15 doi, 10.1128/MCB00857-13. PMC 3,911,281. Retrieved November 14, 2015. Walsh, J.G., Muir of Dar and Power C. Inflammasomes in the CNS. Nature Reverent Neurosi. 15, 84 €97 doi, 10.1038/NRN3638. Retrieved November 14, 2015. Schroeder Kate and Scott Jurg. The Inflammasomes. Cell 140, 821 €832 doi, 10.1016/JCell. 2010.01.040. Retrieved November 14, 2015. Zhao S. and Ting J.P. Inflammasome-associated nucleotide binding domain, leucine-rich repeat proteins and inflammatory diseases. J. Immunol 183, 7623 €7629. doi, 10.4049/Jimmunol 09022425. PMID 20,007,570. Retrieved November 14, 2015. Stuza, Golenbach DT, Lapsi Inflammasomes, Too Big to Miss. J. Clin Invest 119, 3502 €3511. doi, 10.1172/JCI40599. PMC 2,786,809. PMID 19,955,661. Retrieved November 14, 2015. Fink S.L., Cooks and B.T. Apoptosis, pyroptosis, and necrosis, mechanistic description of dead and dying eukaryotic cells. Infect. Immune. 73, 1907 €1916. doi, 10.1128/IAI. 73.4.1907-1916.2005. PMC 1,087,413. PMID 15,784,530. Retrieved November 14, 2015. Martin and F. and Scott Jurg. NLRs join TLRs as innate sensors of pathogens. Trends Immunol 26, 447 €54 doi, 10.1016/JCell. 2010.01.040. PMID 15,967,716. Retrieved November 14, 2015. References